Yeah, Chris and I were just talking about it, how every dollar invested in NIH, what a return on that investment that we get for that, and hopefully we can work that out in the Congress to do a better job appropriating more dollars to that cause. You know, being from Texas, we're always kind of Texas proud, and one of the proudest institutions that we have is a place called MD Anderson, and I would argue one of the best cancer facilities in the world, where people come from all over the world to get treated, and they save so many lives. And so many friends of ours that we have admitted to MD Anderson that they have saved their lives. And we have a great deal of gratitude, and I know Linda serves on the Board of Visitors at MD Anderson, so we're very invested in this. But somebody along the way that we got to know quite well, I would say professionally, but also as a friend, is the president of MD Anderson, Dr. DePinho. I can't tell you how proud I am to have him here today to speak to you about some exciting new clinical trials, but also to expand on the issue that I touched on earlier, and that is the compassionate use issue as well. His bio is very long and illustrious and distinguished. I need not have to read through all of that other than to say he's one of the most admired and respected healthcare physicians in the country. Dr. DePinho, thank you.
Well, I am here as the president of MD Anderson. I have the honor and privilege of being so. I'm a physician, I'm a scientist, but most importantly, I'm a parent. I have 3 young children. They're healthy, and the unimaginable has not happened to them, and I thank God every day for that. So this is a very important issue for me personally, as well as for the institution that I lead and the field that I'm part of. I wanted to tell you a little bit about MD Anderson so you have the perspective, you understand where the context is of the comments that I'll be making. We serve an enormous number of patients. Last year we took care of 122,000 patients. We have a national network that's influenced the care of almost an equal number. Importantly, we have 30 sister institutions in 23 countries. So we lead the largest cancer-fighting network in the world. And that allows us to really get visibility throughout the United States and around the world of the issues that are facing individuals with cancer, including those with pediatric malignancies. We have 20,000 employees with a singular focus. And about 12,000 of those individuals are frontline in terms of taking care of patients with cancer. We are a research powerhouse. Our expenditures exceed $700 million. We're #1 in grants from the National Cancer Institute of any institution in the nation. We have over 1,000 clinical trials ongoing. And of the last 71 FDA-approved drugs, MD Anderson was responsible for leading the trials of 1/3. Again, our experience in understanding the issues with respect to going from discovery to clinical endpoints that matter for patients is the perspective that we have. And our size enables us to deliver multidisciplinary care, bring technology to bear on patients with cancer. We're very proud of the fact that we have a children's cancer hospital that's embedded within the nation's largest freestanding cancer center. It consists of 3 parts: the George Foreman Pediatric and Adolescent Inpatient Unit. This is a state-of-the-art facility that was recently constructed and renovated. It also includes an accredited school. So it's important to appreciate that cancer is not simply about genes and cells. It occurs in a child, in a family, and we want to make sure that the education of these kids continue, and we have an accredited school embedded within our cancer hospital so that children can continue to learn. We also have the Robin Bush Child and Adolescent Clinic. This enables children to come in and out of the hospital and continue their lives as seamlessly as possible. And then Kim's Place, which provides young adults with the distraction so that they can get their minds off of cancer. So we do a lot of things to support not just the medical condition of children, but the context in which those diseases occur to support them and their families. So there are many opportunities. It's important to appreciate that pediatric cancers are a rare disease, and increasingly so as we have a molecular understanding of cancer in general. That same argument could be made for many adult cancers as well. Limited numbers in children with a specific tumor type, and the recruitment for clinical trials, as we all know, is extremely challenging and lengthy. The exorbitant cost that typically goes into creating a drug for adult conditions is prohibitive from a market standpoint because the market size isn't there, as we know. So this is a special challenge in pediatric oncology drug development. And then, as we heard, children do have unique developmentally specific differences, issues relating to toxicity, metabolism of drugs, different formulations, different diseases, different biology are important. And the point was already made. It's critical to understand that a drug that may have had toxicity in adults may not have any toxicity in children, and vice versa. And we have to be mindful of the fact that as we begin to understand the very molecular nature of a disease, that if we do have a drug that is targeted to that disease, we have to be in a position to be able to deploy it with the right kind of framework. So with respect to oncology drugs and children, most drugs used for childhood cancer are not approved in children. New drugs that come to market, if new drugs are to come to market, before we have that data that tells you about the half-life or the metabolism of a drug specific to children, there's either risk of increased toxicity on one hand or less efficacy on the other because of those differences in metabolism of the drug and so on. So this is an important issue to keep in mind as we undergo this dialogue of solutions that are going to be needed for the development of drugs in children. And again, toxicities may be different based on the differences in pediatric metabolism and also varies with developmental stage. It's important to appreciate also that the same target that may exist in an adult cancer, the same mutation that may be resident in pediatric cancer, when you have a drug that's directed against that target, it may have very different efficacy as well. It may work great in pediatric malignancy and not as effectively in adult cancers. The bottom line is you actually have to test the drug. We need that access for drugs so that we can advance the health of children with cancer. So there is a greater belief in the community that children should not be subjected to risk, but this also results in delayed access to new therapies. And in the setting of very poor risk cancers, we essentially have to act. We have to act now. If you've got months or a few years to live, and there's a clinical trial that would need to unfold over a 15-year period, that just doesn't add up. So the ultimate toxicity, in my view, is death. And we have to manage the challenges along with the realities and do the right thing for our children. So smart clinical trials and illuminating novel therapeutic opportunities was touched on by Dr. Collins. It's important to appreciate when you saw those graphs that for certain cancers we have a very poor record of success. We have some successes and we talk about them, but there are many children that are dying of this disease, and approximately a quarter or so of pediatric malignancies will recur. And those children that tend to recur are the ones that end up getting into trouble. Now, I want to tell you about one of our sister institutions. And the data that I'm going to show you, this is the first time that this data is being presented. It's incredibly exciting, and it clearly illuminates the fact that we're on a new path towards really making an impact on pediatric malignancies. So this comes from the Germany Cancer Center, one of our 30 sister institutions. They treat all children in protocols with pediatric oncologists, allowing for the recruitment of some 2,000 cases of pediatric cancers that occur throughout Germany. And about a quarter to a third of those children end up getting those recurrences. So at the time of relapse, a second biopsy is done, and there is very deep genomic analysis that I won't get into, but it's very complex, very deep, very costly, and to do this in the context of their very large genome sequencing facility. So this molecular profiling was done for two reasons. One is to just learn more about what is the genomic underpinnings, the molecular underpinnings of disease recurrence or resistance, but then the other is to identify targets for novel drugs which could be added to the usually unsuccessful standard treatment in this experimental trial setting. So there, as I mentioned, about 2,000 cases annually, about 600 develop recurrent disease, and in the first 50 cases with this very deep analysis, half of the children had actionable mutations. They had alterations for which there were drugs that available that could be applied to those specific cases. Now, you can argue that the experiment hasn't quite been done yet, and we don't yet know if now the application of those drugs to those specific individuals are going to have an impact. But the science on this has already been done in a pioneering study that was done at MD Anderson more than a half a dozen years ago, where we did phase 1 clinical trials, where typically what one does is to just enlist patients who are failing standard of care into a phase 1 clinical trial to look for the right dose, the right schedule, and to assess toxicities. In those cases, about 5%, 1 in 20 individuals, also have a response that signals some level of efficacy. So what we did was to genomically profile those individuals. At the time, it was only 11 genes. This was a long time ago. And that then shepherded half of the patients who had actionable mutations into specific clinical trials, as opposed to randomly being shepherded towards other clinical trials. Those that were brought into the clinical trials that were molecularly informed had a 27% response rate, as opposed to a 5% response rate. So that's proof positive that our molecular insights that we get on an individual level, even with those elemental analyses, and this is far more sophisticated, provides us with an opportunity to match the right drug with the right patient based on the molecular efforts. We at MD Anderson have teamed up with a foundation, Triumph Over Kids Cancer, and we're going to be launching an effort to do deep sequencing on children that recur initially with osteosarcoma, and then we're going to be moving very aggressively in this area and moving forward. And we're going to be collaborating with others around the world, with our sister institutions, to try to scale and bring this up on a very, very high level. The important thing here from a policy standpoint is that this is where the science is absolutely going, and it's going to happen very quickly. So what we have to do is, now that we have that information, we have to be able then to have access to the medicines so that we can apply it to these children who have these actionable mutations. So our challenge here is to do the right thing and bring those stakeholders together so that we can align the science, the regulatory, and facilitate this in a way that really is going to save lives. Because the current constructs are not going to be as efficient as needed in order to take advantage of this truly revolutionary time in the science of cancer medicine. So I want to emphasize that Congress and the FDA have done a great deal. There are many laws that have been passed that have really shown tremendous impact, have advanced the field, and I want to congratulate our congressional leaders for their wisdom in really providing improvement in public health, particularly as it stands for children. And of course, the Creating Hope Act Congressman McCall, this is really, we're just, the level of admiration that you've gained from people in the state of Texas, families and so on, is really beyond words. And I want to thank you for your leadership and really helping drive this through, and Linda as well. You really have made a difference, and it's important to appreciate that this act will save lives. There will people be alive and grow up to be adults and grandparents because of what you've done, and I just want to thank you on behalf of the patients and the families that we serve. It's a tremendous, tremendous advance for all of us. So some policy considerations. So we know the PREA, the requirement that provided the FDA with the authority to require studies of drugs in children, and if adult indication occurs in children. Now, it's important to appreciate that this does not provide much support for tumors only occurring in children. Revisions to PREA that mandate the consideration of molecular targets as a criterion for pediatric testing could bring new agents to children, and that would be very beneficial for the agency to consider. So this is a consideration for us. With respect to compassionate use, it's important to appreciate that many Americans are confronted with this daunting decision when their standard therapies fail, and that's true for in all cancers. 2/3 of patients do very well with standard of care, but for a third of patients, standard of care is clinical trials, and they need to have access to both clinical trials, and if they're not enlisted in a clinical trial and fortunate enough to be at an institution like MD Anderson, we need to facilitate the access of patients so that they can have these potentially life-saving drugs. So this is critically important for diseases like cancer, and providers and caregivers for these patients are truly committed, as we are at MD Anderson, to searching for these life-saving options. So experimental or novel therapies can offer hope for cure, extension, and better quality of life. You know, when I think about those melanoma patients, and we talked about immunotherapy, I know a patient who's alive 13 years later as a result of Jim Allison's pioneering work